So we've spent quite a bit of time looking at this electrical signal in our axons and neurons and how that signal can flow. And that action potential is moving 100 meters per second down an entire meter of your body, some of the longest neurons in our body. That's a way to signal. But we have other chemical signalers that take a message from the outside of the cell, bring it into the cell to cause the cell to change its behavior. And again, we're going to see it begins with a receptor and a ligand. So in our illustration here, we've got the green triangle, the ligand binding to its receptor. That causes this whole cascade of molecular changes. That's eventually going to result in the release of calcium into the cytoplasm. Calcium activates another second messenger. In this case, that's going to be PKC, phosphokinase, that's going to add a phosphate. So do you see the stack of dominoes? That's what a lot of people compare this to. You knock over the first domino, which is the binding of the ligand to the receptor. Each successive domino is going to continue to fall until we arrive at the end point. And the end point in this very simplistic illustration is going to be the phosphorylation of diacylglycerol, DAG. And that's going to help start the process all over again as we see in our illustration. So do you understand the cascading? Signal transduction and often second messenger signaling is because where's the first message? The ligand. All the rest are secondary to the binding of the ligand to the receptor. But notice what else is happening. The information's on the outside, right? In the extracellular space. What's happening here on the second messengers, that's inside the cell. So again, the signal transduction and second messengers gets the signal from the outside to the inside of the cell. The other great thing that's happening when we bring this in, and in our first step, we're activating G proteins. Those G proteins are not just going to activate one phospholipase C, they're going to activate several. And each one of those is going to go downstream and do. So we're going to amplify that. One ligand is going to be amplified many-fold. So the signal is going to be louder, and it's going to go more rapidly inside the cell as we activate all of the downstream partners to get to the endpoint. You see how that will happen? One ligand, and even though in our illustration you just see one, it's going to repeat that many times and amplify and spread and diverge the signal. Don't panic. I could see it in your face already. You do not have to memorize this slide. This slide is just to give you an idea of the complexity of all the different messaging pathways that can happen inside of a cell. And in fact, a researcher could spend his entire career researching one of these. So we're not going to cover all of these in that extreme detail in this course, but we are going to begin to take them one at a time. So again, based on our relationship with receptors, the ligand-receptor relationship. Ligands, small chemicals, the messengers that bind to the receptors. And we've already seen our second messengers. Those are the relay proteins, the relay chemicals inside of the cell that are initiated into motion by binding of the ligand to the receptor. I think another way that you can think of the amplification of the signal, have you ever told a friend something in confidence, and then that friend ended up telling like 4 or 5 people? Then each one of those told 4 or 5 people, and the message got spread really, really quickly. Well, those were the second messengers, and unfortunately, that's real hard to undo once you spread that stuff out. Second messenger signaling, once you start it, it's not going to stop until it gets to the end, almost like an action potential. So chemical signals, we can have hormones. What is a hormone, by the way? We've talked about neurotransmitters, chemicals that are secreted from neurons into space. And I said epinephrine, while it's a catecholamine, it's not a neurotransmitter because it's, number one, not produced by a neuron, but number two, hormones are transported in the bloodstream. And so adrenaline, it can flood through your body really quickly. When you almost get hit by a car at the intersection, it does not take your heart long to speed up. That's adrenaline transported in your bloodstream. We can also have chemicals that are just local in the environment. A cell can actually affect itself. It can have this autocrine regulation where it produces something that binds back to its own receptors. It's like paying itself, but it can also affect the other neighbors and the cells that are surrounding it in that local environment. So with our receptors that we have on our cells, in this case we got our ligand, just a generic looking, what is that going to be, a hexagonal shape, and there we've got our hexagonal receptor. So we've got specificity of binding. A square is not going to fit in there. A triangle is not going to fit in there. Only the correct molecule. So again, when we think of enzyme substrate, that perfect fit, you're going to have that perfect fit with receptors and ligands. So, not surprising that the shape is going to be dictated by the amino acid sequences. And not surprising that this interaction between the ligand and the receptor is facilitated through non-covalent bonds. Principally what kind? Hydrogen. Because you don't want a ligand just to get into the receptor and get stuck. No. You want it to be able to bind, activate, and then come off. And the binding and the stimulating of receptors often going to be controlled and regulated by concentration. If you have a lot of ligand out here, that thing's going to get turned on and maybe continually turned on. If you just got one of these out here, just the accidental collisions, it's going to be much slower. So concentration is going to play a huge role in this process. The other factor in signaling is going to be how strong a receptor can bind to the ligand. This is referred to as affinity. Think of magnets. Have you ever played with magnets before? Like refrigerator magnets you may have, or some little toy magnet. Yeah, that's one thing. Have you ever got a hold of one of these rare earth magnets? Like one of these magnets that they use to pick up the roofing nails, you just kind of drag it behind you. When a nail gets on that thing, you can almost not get it off. It's so strong. So affinity, a high affinity ligand receptor relationship, man, you're going to bind that ligand extremely easily, and it's only going to take low concentrations before you're really going to stimulate that receptor because it's going to be there a while. Versus a low affinity ligand receptor interaction, it's not going to bind that easily, and it's going to be real easy to bind and pop back off. So it's going to take a lot higher concentration to signal the same for a low ligand relationship versus what would happen with a high affinity ligand relationship. Does that make sense? This really relates to how long that ligand is going to be in the binding site. To mimic the same thing, you're going to have a lot more popping in and out to mimic the same kind of binding you have there. So the way that this is reported, how strong a ligand binds to the receptor is referred to as affinity, and it's reported as the dissociation constant. So you look at the concentration of your ligand that's needed for half of the receptors to be occupied and stimulated to give us this dissociation constant. And so when we have high affinity receptor ligand relationships, it's going to have a low dissociation constant, meaning it's not going to take a high concentration to get a really good signal. Versus a low affinity relationship, the concentration is going to have to be much higher to get the same intensity of signal, simply because of the on-off rate of the ligand binding to its receptor.